that was enjoyed by all. I'm happy to introduce our moderator for this session, Madison Sturba. We're going to be talking about horizon scanning this afternoon for the next 2 sessions. So stay tuned for that. I'll pass it over to Maddie to take it.
Thanks, Mackenzie. Hi, everybody. Welcome to part 1 of Horizon Scanning. I hope you're just as excited as I am to hear from our speakers today. Just a quick housekeeping note, if you have questions at any time, please add them in the Q&A box and I'll read them out to our speakers at the end of their talk. So with that, let's dive right in with our first speaker. I'm excited to introduce Alanna Richman from the Pennsylvania State University to talk all about spotted lanternfly. You can share your screen and get started whenever you're ready, Alanna.
All right, thank you. Can you see my screen?
Yeah, that looks great.
Okay, thank you. Sorry, I couldn't tell. All right. Hi, my name is Alanna. I'm a first-year ecology student at Penn State University, and I'm going to be talking about some of the work I did on my master's thesis at SUNY Albany. So I looked at the assessment of spatial distribution patterns and infestation risk over time in spotted lanternfly, which I'll just be saying SLF, throughout New York State. Hold on. Okay, there we go. I'd just like to quickly acknowledge the members of my thesis committee at UAlbany for their guidance, as well as the New York State Department of Agriculture and Markets, where I worked at the time and got a lot of this data, as well as my current PhD advisor at Penn State. Oops. Hold on, I'm having a little bit of technical difficulties. Okay. All right. So just a quick introduction, spotted lanternfly SLF were introduced to Pennsylvania State in Berks County from Asia in 2014. And they have since spread to many states throughout the United States, but to New York specifically in 2020 on Staten Island. Little is known about SLF spread in biology in New York State specifically. And the preferred host plants for SLF are tree of heaven, which is very important for nymph growth, as well as grapevines, fruit trees, maples, willows, and a bunch of other plants, but those are kind of like the main ones. So SLF invasion has been significantly associated with short and long distance dispersal. So an adult SLF can move about 3 to 4 miles within their lifetime, but the spread patterns we're seeing are not well explained by that. So human-mediated dispersal has showed significance. Basically, SLF will, normally they lay their egg masses on tree bark, but a lot of times they'll lay on inanimate objects and on vehicles, and these will either get transported by vehicle or just be on the vehicle itself, and then they'll hatch in a new location. So a lot of times we see them popping up along railways and like major highways, distribution centers, etc. Some of the main concerns regarding SLF, so obviously ecological, a lot of studies are focused on the agricultural side of them as they are a huge agricultural pest, but there's a few studies that show ecological issues like native like maple trees and such. Also honeydew accumulation is so that's what they excrete after they eat the sugars in trees, and then that allows for the growth of sooty mold, which causes a blockage of photosynthesis in understory trees. Climate change, as we know, is a huge issue regarding invasive species. It has been shown that SLF can still survive even if like favorable and unfavorable temperatures vary within like say like a winter season. So that can be an issue even in cold climates where we don't think they're going to establish. But if we have some warm days due to climate change, they can still establish. And then economically, so in Pennsylvania State specifically, it's been estimated that $11 million have been lost due to like agricultural reasons regarding SLF, so fruit trees, berries, grapes, tree nuts, etc. And New York, as a neighboring state, has a lot of similar agricultural industries. Okay, so to start off my study, what I did was I collected SLF presence data, which accumulated to 2,899 points. These were collected from the New York State Department of Agriculture, which I worked for, and collected a lot of this data. However, this was an accumulation of data collected by many inspectors throughout the state. I also used verified IMAP invasive data, which were usually verified by people in the agency I worked for anyways. All maps and analyses were done using ArcGIS Pro and MGWR 2.2. And then I used additional sources listed above for some of the other things I looked at. So for my methods, I did a spatial autocorrelation hotspot analysis to look at dispersal patterns, a space-time cube visualization to look at spatial temporal trends. I identified areas of high concern using a weighted overlay. And I used a multi-scale geographically weighted regression to see if there was a relationship between SLF counts per municipality and any other spatial variables. All right, so this map is just showing the positive sites that I use in this study. They exhibit a significantly clustered pattern. As you can see, this is mainly in like downstate New York, and then pockets going to like major cities in upstate New York. So like Albany area, Buffalo, Rochester, Syracuse, etc. What I did next was I counted each of the positive sites per municipality in the state to get count data, and that made it easier for some of the other assessments. So the map on the left is just showing that distribution. And again, like you can see that the counts are higher towards like downstate New York. And then I used a hotspot analysis, which is essentially just looking at where there are areas of high counts near other areas of high counts. So this is mainly showing downstate, like Western Long Island area is where the hotspots are. This space-time cube visualization shows a significant downward trend near Staten Island. So that's the little blue squares. So this could signify that there's kind of been a population like stabilization or decrease, which would make sense because that is where the spotted lanternflies were first found in the state. And then we see significant upward trends, which are in red, kind of like sporadically spread across the state, showing that the insects have spread in like all directions from Staten Island. And then this was the concern level map I made. So I made this with the intention to help the agency I worked for, as well as if it could help any others. What I did here was I took agricultural district data and I made transportation vector buffers that were 3.5 miles long due to what we know adult SLF can move in their lifetime. And this is the map that resulted. I did 5 concern level classes. And then 86.3 of all these positive sightings I used were located in what I designated as the mild concern area in the middle. So it is a good thing there's not too many in the very high risk areas. And the results of my multi-weighted geographic regression, so this resulted in an R-squared value of 0.177, which is a bit low. However, I did get some interesting insights from this study. The independent variables that showed significant p-values included the counts of tree of heaven, square miles of railroad buffer, and square miles of road buffer, and this is all per municipality. So this is a map of how the model explained the municipalities on the local level. So there's like greater range here. So you can see in the darker green where like what areas were better explained by this model than others. So the New York City area, as well as some of the Hudson Valley, seems to have it explained a bit better by this model. And then when looking at the three independent variables that showed significance, so Tree of Heaven showed strong local significance in all but one municipality, which is interesting, and I'll talk about further because this variable had global and local significance. Railroad buffers actually show no local significance, so it only showed significance on the global scale. And road buffers showed significance, local significance in 11 municipalities. So like Eastern Long Island and like some of the Hudson Valley are the areas where it seemed to matter locally. And the main takeaways from this study showed that despite the global results only explaining 17.1% of positive SLF sites, locally the results differ in strength, so they can be taken with more significance and serious in certain areas. SLF distribution is highly clustered, especially in these urbanized areas. Downward spatial temporal trend near Staten Island may indicate that there is a population stabilization or decrease going on there as the insects spread further into the state. Significant upward trends scattered across the state going out from Staten Island show like the movement patterns. And then the identification of areas of high to low concern, like those 5 classes I made, could be used to guide future SLF survey and management plans. And I think this would be particularly helpful with agencies that have very limited resources or staff, so you can kind of pinpoint where you want to send people to do surveys and where you want to focus your money. My recommendations based on what I found was the strong influence of Tree of Heaven in both the global and local regressions suggest there should be strong focus placed on the management and mitigation in areas with these dense populations. So you can kind of narrow it down to areas of high concern, and then, okay, where are there are a lot of Tree of Heaven, even Tree of Heaven is very widespread, but using different sources of data, you can look at that. Areas, so yeah, again, doing that. And then what I also wanted to emphasize was full-scale ecological threats posed by SLF need to be taken into further consideration. So from what I've seen, most of the studies regarding SLF have an agricultural focus, which is very valid as they, especially in New York, are a problem for orchard industries. However, there is a severe ecological component, and I think that just looking for them in our native forest, as opposed to just agriculture, is important as well. And here's some of my references from the study, and I'd be glad to answer any questions.
Thank you so much, Alanna. That was really great. SLF is a really hot topic here in Canada and Ontario, so it's always really great to see the research going on and what's happening in the state. So yeah, I really appreciate you joining us. We do have a couple of questions coming in. If you do have questions, please pop them in the chat. We have a few minutes. The first one is, what was the last year of records included in your analysis?
Oh, yes. So the years I used were from 2021 to 2023.
Perfect. Thank you. There's a question here. Could the downward trend of SLF in Staten Island be due to different surveying pressure? Once we know something is widespread in an area, we don't tend to look for it as hard or get as many public reports. Were surveys consistent from year to year?
Yeah, I agree with that. So some of the limitations to the study were how the data is reported and such. So like in New York, they hit a point at a certain year where they kind of were putting less emphasis on looking in the New York City boroughs just because like they know it's there. So that could definitely be a factor as well. But yeah, my intent with this study was to just using the data available, kind of get a preliminary idea of what's going on with the spread of this insect in New York State.
Definitely. Yeah, that's really helpful. Another question here came in through the chat. Specific to Staten Island reporting, was the request for residents to not report in known areas in Metro NYC and saturation of reporting from those areas considered when calculating the reduced population on Staten Island?
Yeah, I do know that they asked residents not to do that with the government agencies. That's why I also added in IMAP invasives data just to help to maybe offset that a little bit, because that can kind of be done by anyone and not necessarily like submitted directly to an agency. But yeah, that was definitely a limiting factor there as well.
Okay, awesome. Good to know. Thank you. I'm not seeing any other questions, but there are some comments. Just good to see you doing the great work and carrying on the fight for SLX. So yeah, it was a really great talk, and thank you so much for joining us.
Thank you.
Awesome. Okay, so I'm happy to introduce our second speaker, Justin Kreller from Carleton University and Nature Conservancy of Canada, who'll be talking about broad-scale simulations to understand terrestrial plant invasions. Thanks so much for joining us today, Justin.
Thank you. Awesome. Okay, I'll share my screen. Yeah, so thanks for having me. As mentioned, I'm a master's student at Carleton University studying invasive plant spread under the Weston Family Conservation Science Fellowship Program at the Nature Conservancy of Canada. At Carleton, I am a part of Dr. Joseph Bennett's lab, which in part develops management optimization tools for biodiversity conservation. And I am co-supervised by Dr. Emma Hudgens, a recent postdoc of the lab and now a leading expert in the study of invasive species spread through anthropogenic means. She is now working at the University of Melbourne in Australia. So to start, a little background on my study. North America has accumulated the most non-native plants over the last 150 years, and this trend is only expected to continue rising in the years to come. So this outlines a need to forecast the harm that these plants are causing to our native ecosystems, both across contexts and across species. My project works off the fact that invasions are primarily human-driven. And so we're working off the assumption that there are shared predictors of spread across species. And if so, there could be large-scale patterns of spread that could be exploited for landscape size modeling and predictions of invasions, especially when monitoring information is limited or there is a new invading species. So the goal of my study was to build it still is, sorry, to build a multi-species terrestrial invasive plant model that is focused on dispersal and based on anthropogenic and environmental variables. And the goal is to provide insight to focus plant invasion management and target our conservation actions to protect existing natural areas and determine potential future protected areas. To achieve this, the first goal of my project was to build a simpler version of the model based on a single species, and then expand that later. So the species that I chose to start with is called dog strangling vine. It is also known under the names of European or pale swallowwort. In French, the name is no less intimidating. It's known as le dent venin de Russie, which means venom master or tamer of Russia. Its scientific name is Vincetoxicum rusticum, and I'll refer to it throughout this presentation as DSV. So DSV is part of the milkweed family. It is native to Eastern Europe and can grow up to 2 meters high. It is a perennial herb, and it is a vine. So what that means is it'll climb up other vegetation, it'll trail along the ground, or it'll even climb up other plants of the same species up to a 2-meter height where it'll fall under its own weight. It has bean-shaped seed pods and small pink-purple flowers that resemble starfish. And it reproduces through root fragments as well as through the 2,400 wind dispersed seeds that it produces per square meter. It was first found in Ontario in the late 1800s, and now today is also found in Quebec, New Brunswick, and Nova Scotia, as I've got illustrated here using data from the Nature Conservancy of Canada, as well as AdMaps and the Global Biodiversity Information Facility. It prefers sunny areas or lightly shaded areas, and it invades ravines, hillsides, fence lines, stream banks, roadsides, utility corridors, as well as prairies, alvars, and tree plantations. Its current distribution was one of the reasons why I chose the SV as a starter species, as it's relatively restricted to a tract of southern Ontario and Quebec, with a few long-distance dispersal events in northern Ontario and the Maritimes. So its impacts, it affects the environment, our society, recreation, like all invasive species do. So to be a little more specific, Dachshund vine tends to grow in very dense patches. So it very easily overruns habitats, such as globally rare alvars found in Ontario, and it displaces the native animals that depend on these habitats. In terms of species at risk, the monarch butterflies misidentify this invasive milkweed plant for native milkweeds when laying its eggs, and the larva can't feed on Dachshund vine as it doesn't provide the proper food source. The larvae die and don't become butterflies. And finally, DSV also shades out native plants and young trees very easily, and the understory has a hard time regenerating as a result. DSV may be toxic to livestock and can short circuit fences, and its interwoven mat of vines are difficult to walk through, so they hinder recreation as well, as they tend to grow trailside. Some of the controls for DSV include active monitoring, as well as mechanical and chemical controls and disposal. In terms of mechanical controls, digging is done to eradicate large patches. But for smaller patches, you can also reduce seed production by mowing, clipping, pulling, removing seed pods, as well as tarping. Finally, spot herbicide is applied to control medium-sized populations as well. Okay, so in terms of methods for my project, I take that occurrence data that I showed you earlier, and as well as a bunch of data on potential drivers of spread, and start by overlaying a grid over this spatial information, and summarize it by each grid cell. So at the top here, you can see I've got occurrence data summarized by grid cell in terms of how long the species has been present in a certain grid cell, darker colors being longer invasion times. So notably around Toronto, Ottawa, and Montreal, with newer invasions around Quebec City, North Ontario, and the Maritimes. And then data on potential drivers of spread on the second row here. So I've got power transmission lines, mean annual radiation measures, as well as the amount of rivers in a given grid cell. So all of my candidate predictor variables for spread are spatial in nature, and some of them, as you may have noticed in the previous slide, are also temporal as well. They have that time dimension. And so Not all data had available data sets for this, but when there was, for example, using census data throughout the years for human population density and household income, as well as some of the environmental variables that I used this to get a temporal dimension to my model. So I've got all this data. How do I find out which one was most useful at predicting spread? So I start with, what I do is I use a regression-like forward stepwise selection procedure, which looks at each of the variables one at a time. So what I do is I start with a constant dispersal model. So a model with no drivers of spread, just a constant dispersal rate, and use that as my base model. And then I enter what I call the model building loop. So what this will do is it'll add each variable one at a time to the base model and evaluate how it does at predicting spread to date. So yeah, like I said, it adds each term sequentially to the model. And then there's these 4 steps here: introduce the species at its initial distribution. So I chose that to be at its distribution in 1960. And then allow the model to simulate the spread, compare that to what's observed today, and then come up with a way to evaluate that comparison. And then what I do is I determine whether that metric of fit is adequate, if it adds enough predictive ability to the model. And if so, that term gets added to the model and the loop continues. Or it exits the loop since the model doesn't add enough predictive ability to the model. And just to go into a little more detail on this point on the model building here. So again, we introduce it where it's first found, and then I use an optimizer in R to come up with a bunch of different simulations. And then the one that does best is retained for that variable. And then, so as you can see here in the graph, R is creating this model, and you can kind of see the predicted curve getting tighter against the observed curve. And then we compare that to what we see, and then the loop continues. So some of my preliminary results, you could see a 4-parameter model here with 2 variables related to the environment and 2 related to human activity. The mean annual radiation and roads variables being retained corroborates the species' preference for sunny areas and roadside ditches. Each variable is also evaluated from the point of view of immigration into a cell, its capacity to bring propagules in, as well as its ability to create spread. So you can see that 3 out of the 4 variables were impacted by immigration. So what does this mean in practical terms? So we can see that the parameter values are negative, and the component of spread is impacted is immigration. So what this means is that the model predicts that DSV persists at sites that have more sunlight, deeper soils, and more roads. And conversely, positive values related to immigration have to do with, they suggest that a DSV disperses to places with higher mean annual temperatures. And finally, this 4-parameter model seems to predict about 35% better than just the constant dispersal model, which is relatively adequate because the spread from Toronto outwards is radial enough, and that's what a constant dispersal rate captures. And so the extra 35% comes from predicting the other more long distant areas. So I've obviously some improvements and limitations to bring. I'm still awaiting a hiking trails data set that I can include from across the country. And some limitations currently include that I use one single grid for the entire simulation. So the effects of spread are all captured at the same spatial scale. And so cross-scale effects are not seen in a single simulation. And not many of the data sets were temporal in nature. And finally, some next steps. So as mentioned, I want to build a multi-species model. So I'll be expanding this model to be fitted to a diversity of species to identify shared drivers of spread. The next step after that will be forecasting future distributions under various socioeconomic and climate change scenarios, with the goal of sharing these dispersal insights and spread forecasts with conservation managers nationwide within the Nature Conservancy and out by making this open access. And then, if time allows, prioritizing protected areas for management based on risk. Lastly, I just want to acknowledge the following organizations for their generous support for my project: the NCC, the Weston Family Foundation, as well as MyTax and NSERC. Thank you.
Thanks so much, Justin. That was really great. Super impressive. If you have any questions for Justin, please pop them in the Q&A box and I can share them. I can get us started with the first question though. You mentioned for your next steps that you want to expand the model to a diversity of species. Are there any species in particular you'd be interested in including?
Yeah, so there are a bunch of species, that I know of around where I am in Ottawa. But I'm trying to get a diverse perspective, right? I'm trying to look across contexts. So what I'm doing is I'm going on the NCC's website, I'm going on Ontario Invasive Plant Council websites, like the Invasive Species Center websites, and I'm compiling a list of what they have as the most invasive plants around Canada or their region. And so basically the ones that are just most popular, I'll be gathering the occurrence data for later on, and then using that in my data set. So for example, Phragmites are a huge one. And then stuff like giant hogweed, which has a lot of, societal effects or like impacts. Out west, there are species, I believe, Scotch brome, a bunch of grasses that affect agriculture. So I'm trying to capture effects from a bunch of areas. So I'm contacting people from conservation agencies and all that. Yeah.
Yeah, and that'd be a lot of work too to try and compile all that information from all those different places, I imagine.
Oh, but it's a lot of fun because I'm discovering new species all the time. Yeah.
Yeah, that's great. We do have a question that came in through the Q&A. Would the correlation between input parameters such as road number and population density have an input on your model performance?
Yeah, especially the environmental variables are highly correlated. So those specifically, I run a correlation test after the first run of the model, and then I get rid of those with higher than 0.8 correlation, just to be a little stricter. And really, the model, the way that it selects each of the variables, it kind of takes the correlation into account in that it won't add variables that don't add much more than a certain delta of improvement to the model. So if roads are highly correlated with hydro corridors, then it won't add the power line to the power line data set to the model because the roads data set was already selected for. Does that make sense?
No, that's great. Thank you so much. This person here, they have solid walls of glossy buckthorn at Copland Forest in Simcoe County. Do you happen to have any models for this species for glossy buckthorn?
So no, not yet. And so when I expand my model, it'll be expanded. When I mean expansion is I'm going to fit the model to a bunch of species. So hopefully to a suite of species with a diversity of life histories. And so the result will be that the model will hopefully show which drivers of spread are most important across all species. So it's not that I'll fit it to a certain species in particular, but I'll fit it to a bunch and then see what the shared drivers are. So are they more related to human activity or the environment? And that kind of insight can then be used for a specific species in the controls in and whatnot.
Oh, awesome. That's really cool. Yeah, thanks so much. I think we'll end it there for questions, but really appreciate your time, Justin.
Thank you.
Okay, so we're excited to have Natalia Castillo-Mendoza from the Institute of Ecology at the National Autonomous University of Mexico, or UNAM, as our last speaker of the session, who will be talking all about ecological niche modeling.
Okay, so you can see my presentation?
It's just loading. Yes, it looks great.
Okay, well, hi everybody. Well, I just want to mention real quick that this is my master's project, which is a segment of a larger one, and I modeled the potential distribution and dispersal route for the moth, while other of my colleagues made the models corresponding to the native species of Opuntia. And from North America and the Caribbean, and later on it's going to be put together. So without further ado, well, Cactoblastis castorum, commonly known as the prickly pear moth, is a native species from South America, which feeds on several species of the genera Opuntia. So I'm going to tell you a little bit about their life cycle. So the moth oviposits on the areola of the thorns of the leaf, and once they hatch, they pierce the epidermis to feed on the non-fibrous tissues of the leaf to continue their development there. And when they reach maturity, the larvae heads towards the nearest ground area to the plant to form their cocoon, given that they use the cacti's debris and loose soil to cover them up for protection. And once they reach adulthood, the moth emerges and flies to a nearby nopal or cacti to search for a mate. And well, it was introduced as a biological control agent from Argentina to Australia in 1925 to control several species of Opuntia from North and South America. And it was later introduced with the same purpose to South Africa, Hawaii, and the Caribbean islands. During its expansion in the Caribbean, it has sharply reduced populations of native Opuntia species. And it was detected in Florida in 1989. And ever since then, it has been considered one of the greatest threats to the biodiversity of this native cacti from North America. So in Mexico, the profits from Opuntia crops amount to about 265 million US dollars. And in the year 2007, the occurrence of Cactoblastis cactorum was first reported at Isla Mujeres, which is over here. This is a small island over here. And by the year 2008, it was eradicated. Nevertheless, it has been categorized as a species with a high probability of entry, given that populations nearby the Mexican border keep growing. And in addition, hurricanes that with trajectories passing over the Caribbean islands infested with the moth impact Mexican territory, entering through the Yucatan Peninsula, as well as some areas of the coast of the Gulf of Mexico. And one way to assess the invasion risk of the moth is through ecological niche modeling and the study of its dispersal dynamics. So, well, in this work, we modeled the potential distribution of the moth using ecological niche models, which estimate the set of environmental conditions where population growth rate of a species is positive. And in the context of invasive species, such tools facilitate monitoring and dispersal predictions. So the research questions are: which geographical areas in North America and the Caribbean are most suitable for the establishment and dispersal of Cactoblastis cactorum? And what are the high probability invasion routes for the moth in Mexico? The methods. So in the first phase of this methodology, we gathered occurrence data of the moth from its native and invaded area, as well as a total of 99 variables at a 30-second resolution, which were bioclimatic and of chemical, physical, and temperature properties of soil. And remembering the life cycle, we used these two types of variables to try to identify if there was any pattern of preference of soil type by the moth to pass their pupal phase there. And in the next phase, the database was thinned and divided in two parts, and one was used for the training and another was used for the test of the models. And all this data is prepared for the next phase, which is the calibration of the niche models. So in this, a sperm correlation was performed, and the predictors whose correlation factor is less than or equals to 0.75 were selected. And we explored two modeling algorithms, which are the minimum volume ellipsoid modeling and Maxent, both with R language packages. And the candidate models pass through statistical and performance filters. And the final selected models are binarized in such way that pixels with the value of 0 represent non-suitability and 1 indicates suitability. This is at a restriction threshold of 5 percentile. And well, for example, this dark green area here tells us that all models agree that this area is suitable for the moth or any species. And as the color goes lighter, the frequency of the models that indicate suitability in those areas also diminish. And so, well, the next, this map which is a consensus map for the selected distribution models, is then normalized to have continuous values between 0 and 1. And it's used in the next phase, which is the estimation of the invasion dynamics, which I'll explain a bit better here. So we have our normalized consensus map, which is binarized at different restrictive thresholds to explore the connectivity of the suitability patches. So for example, in the case of our study, we binarized it to 100 different thresholds that go from the minimum training presence to the 10 percentile. Well, we used the cellular automaton kernel for the simulation. And well, for example, to illustrate, if we would have these 3 restrictive thresholds and this little triangle indicating invasion of the pixel, the purple pixel indicates no suitability, and the yellow patches indicate suitability for the moth. So at the final time of our simulation, we'll see that all these highly connected areas going to be invaded. Well, on the more restrictive thresholds, there's going to be areas that are not going to be invaded given the lack of the connectivity of the patches. So adding these dispersal simulation scenarios, we get the simulation consensus map, which is this one. And now on purple, we see pixels that are not invaded. And as the color changes from purple to yellow, the frequency in which the pixels were invaded increases. So this area, this yellow area here, would be the high probability invasion route. And we explored 3 dispersal scenarios, which are short, mid, and long distances, and 2 initial points. One, which is from the nearest occurrence data from to the Mexican border, which is at Rockport, Texas, and one at the southeast continental area, closest to Isla Mujeres, where the moth was in 2007. So let's go to the results. So this is our final data and database, and I show you here in purple occurrences from the native region of the moth, and on green occurrences from the invaded areas. So, well, we obtained more than 4 million candidate models, from which 2,075 were selected. And so this is our consensus normalized model. And in red, I show you the areas that are suitable according to our models, suitable for the moth. And we can see that it also predicts where the moth was and still is. And making a zoom for Mexico, we can see that the most suitable areas in Mexico for the moth to establish and disperse is along the coast of the Gulf of Mexico. And well, we can also see that it predicts where it is currently at Florida. And these brown dots are occurrences of Opuntia species native from North America and some from the Caribbean. And well, the results for our dispersal simulations, we can see over here on the simulation that this a green dark area is the high probability dispersal route. And well, this is the same map in different colors just to make it clearer. So 90 to 100% of the models invade this purple area. So our purpose was just to estimate this high probability dispersal route, but we made a very rough estimation of the time it would take the moth to disperse from this starting point to this environmental barrier, which is the volcanic transversal system. And sorry, it would take about the moth to reach this place about 12 years. And well, we made calculations based on the maximum dispersal capacity of the moth reported by Johnson and Stirling, which is 75 kilometers per year. And from the southeast starting point, we can see that, well, only 90 to 100% of the models invade this little area under the star. And well, it would take the moth to reach, more or less, to reach Rockport, Texas, about 26 years. And well, our conclusions are that for the ecological niche modeling, that the most suitable area for the moth to establish is along the coast of the Gulf of Mexico, and that there are less suitable conditions in the Yucatan Peninsula for the establishment and dispersal of the moth. And for our dynamic dispersal routes, that the highest probability route is from the Texas population. And well, I finally want to thank my work team and my supervisor and the institutions that financed this project. And well, I also want to thank you for your attention and presence.
Thank you so much, Natalia. That was really great. If you
Thank you.
have, yeah, if you have any questions for our speaker, please pop them in the Q&A box. I really didn't know anything about cactus moth before this, so this was a really great learning opportunity. Thank you.
Thanks.
Yeah, let's see. I don't see any questions coming in just yet, but just for our own information, are there any ways that we could help prevent the spread of this moth when we're visiting or living in those areas where it's been found or where it could potentially establish?
Well, I think one way would be to prevent the transportation of Opuntia species, which is one. And yeah, I mean, it was also translated, I mean, transported to Florida illegally because they wanted to control, well, they wanted to control species that were native. But yeah, I think that would be just one.
Yeah, okay, yeah, that's great to know. Thank you. Okay, I don't see any other questions coming in through the chat here. But if you do have any questions for Natalia, we do have, we've been posting a link to a doc in the chat, and it'll have her email in there, so you can go ahead and reach out directly. Okay, thank you so much, Natalia. Really, oh, wait, just kidding. I see one that just came in, a question for you here. How might climate change affect your models?
Well, yeah, well, we can, there are models that do include these environmental changes. And so, yeah, maybe, I don't know, maybe the areas that would be suitable for the moth would change from the geographical space. But for now, it's all I can say. But yeah, it would be nice to add it up to the analysis, which perhaps we'll do later on.
Definitely. That's awesome. Thank you. Another question here. Southwestern Ontario has a native Optuna up to, the cactus. What temperatures does this moth survive?
Well, the lesser, I think that the least it takes is around 18 degrees Celsius. And at 35 degrees Celsius, it starts to decay. Population starts to decay.
Okay. Yeah, that's really great to know. Thank you. Okay. So that's, I think, it for questions now.
Okay.
But thank you so much, Natalia. Really appreciate your time and for you joining us.
Thank you. Thank you very much.
So, okay, so that's a wrap on part 1 of Horizon Scanning. Thanks so much to all of our speakers for such an informative session. I just wanted to quickly highlight our municipal community of practice, which is a great way to connect about invasive species issues and incoming threats. The COP is a peer-to-peer space that encourages collaboration of those who work in municipalities, conservation authorities, and Indigenous communities involved in invasive species management at the local level. So you can sign up on our website if you're interested in that. We'll take a little break now. We'll regroup at 2:40 for Horizon Scanning Part 2. Thanks so much and see you there.